This is ChatGPT's interpretation of what a cell. This is ChatGPT's interpretation of what a cell is. And what I like is that DNA is central, right? So this is DNA is in every cell, and I'm talking about the DNA is in all the cells of your body, and it's broken up into chunks, and each of those chunks is called a gene, right? So when we talk about a gene, we're talking about a very small portion of the DNA. You have roughly different numbers here, depending on how you give it, but about 25,000 different genes. And what genes do is they're kind of the instruction manual. The DNA sits in the middle of the cell, and it tells the cell how to make various types of products. And that product is the protein. And the protein goes out in the cell and does structural stuff, it does enzymatic stuff, it does everything else. So that's kind of the very this was discovered in like the early 1900s to the 1960s, but it's called the central dogma of cellular biology. So you just learned 100 years of research. Okay, so taking a step back and thinking about genetic testing, I'm going to do more of try to do more principles and then apply it specifically to Pheochromocytoma. And how do you decide whether or not you should do a genetic test? And ideally, your genetic test has high utility, that it changes the course of what you're going to do. And the opposite extreme is you learn something, and I don't want to say that there can't be utility here, but there's not, but in some cases you can't do anything. So you just learn information that's hard to deal with and non-actionable. So that's a less ideal. Some people really just want to know, and that's really valuable. So I don't want to take away that. But this is kind of the initial framework. And I think I place PPGL on this high to medium list. And it's on the medium side because we'll learn. It depends upon which gene, but most genes are high, but sometimes it's not as high. Okay, so this is kind of looking at how do we prioritize which genes and whether or not you say yes or no. So if the condition has a higher morbidity, there's problems, complications that you want to prevent, obviously that's more reason to do it. If there's an effective but imperfect treatment, I think this applies to Pheochromocytoma, which is that we find things early, yeah, we can do something, we're pretty good. If we find things late, it's imperfect, right? With that, so there is a very high utility to finding things early. Is the test predictive? It doesn't tell you whether or not you're likely to have a condition. And here, this is where we lose a little bit of traction. The high prevalence, which I'll talk about, or high penetrance is more useful. Lower penetrance a little less useful. We'll talk about what penetrance is in a second. And then high cost of screening. So yes, we can't do full body MRIs and PET scans on everybody, right? We have to figure out who are the folks that need directed care. Okay. I think that's kind of enough for kind of thinking about that. So when we think about why should I undergo genetic testing, what are the advantages? What are the, you guys are here, so you probably would choose yes, but not saying that you have. But the utility is that if you have a gene, it can tell you something about how the tumor you have is likely to behave. Is it likely to be a more aggressive or less aggressive one? It also tells you whether or not there might be other conditions we should be looking for, right? So for instance, for SDHB, the succinct dehydrogenase B gene, we talk a lot about the Pheochromocytoma, but remember there's kidney cell, renal cell carcinoma, there's GIST, and we don't want to leave those out, right? That's one of the advantages of knowing what gene, what are the specific other risks we should be thinking about. And then the other thing is family members, right? So you both the advantage of finding out who's at risk, and then the advantage of finding out who's not. So the exoneration side, which often is kind of overlooked, but it's very important, particularly as parents, mothers, and fathers. Okay, I think this you've probably heard a billion times, but I'll just reiterate that universal testing for all Pheochromocytomas, everyone should have genetic testing. That's been more, but we recommend counseling and decision-making, co-decision-making, right? It's the individual, it's everyone's right to choose whether or not they want to do it. I would say for genetic testing hasn't been proven to be useful for all the genes that we test. So if you do a genetic test, oftentimes or sometimes you'll get back a gene where we're doing the best we can, but we don't know if it's useful. And then as also been emphasized, there's no strategy that has been proven to be the best one. So we follow a protocol, which are kind of what we agree upon. But that protocol hasn't been compared to some other protocols. So this is really the best one. It's just kind of what we do. Okay, I was specifically told not to do a Kaplan-Meier. This is a Kaplan-Meier. I'm doing it anyway because I think this is really the central purpose of genetic testing encapsulated in a single slide. Okay, so this group here, this is looking at time in years. So we're at 25 years. So the overall message isn't so bad, but in the folks who have SDHB gene, but they were discovered because they had a tumor, at 25 years, roughly 40% of the folks are no longer alive. If you compare that to their family members, so you did genetic testing and then you figured out someone was affected, at 25 years, you're up at 90, 95, 100% survival, right? So it is a marked difference in how the people, the family members do compared to the individual who was found late, right? So this is why you want to do genetic testing. This is the strongest evidence we have for like my existence. So that's why. Okay, so I also do, because we do genetics counseling, I think it's important to consider the principles of genetics, that it is your right, every individual right, to choose whether or not they want to do this, and they shouldn't feel pressure. They can have all the evidence that there is and say it's not right for me, and I'm okay with that. And then children are a special case, right? Because often we make the decision for them, and we recommend in this condition genetic testing. The conversation is different for different genes, but it's typically age 8 to 10 is where we start to say that's about the age that we want to do it. And then this is really hard for a lot of folks. Their sibling doesn't want to do it. And they have the burden of the knowledge and what they should do. But remember that they have the same autonomy that you have. Beneficence enough. Okay, so this is the idea that we want to do something good and we don't want to do harm. And so when we talk about genetic testing, it's important not only to emphasize the benefits, but also it can be hard to find out that you have a gene and then you have to tell other people. You can find out information you didn't want to know. You tested all these genes, but you got another one. Now what? And then there's a benefit to screening. I don't get me wrong, I'm in favor of it. But every year you're going through these scans and these, it's a burden, right? So I think it's important to have the conversation that it's not all roses. There is benefit, but there is some burden of knownness. And then, of course, the principle of justice, that this should be available to everyone and it should be equal access and coverage. This is a big issue across different locations and cultures, and so I'm not going to go into detail here. And confidentiality which is that you have the right to have this information and have it not shared. The conflict here again comes back to family, because there's this balance between whether or not you have a duty to tell them, meaning if I know that my sibling is at risk for a condition, am I ethically obligated to do that? That's each individual's decision to decide. As a physician, most law says I have to advise you to say you should share this. I don't have to enforce it and go and tell your sibling that, oh, this is in your family. And that's based upon this called the limited duty to warn, because it's not an imminent danger, right? It's a long-term danger. So the doctors don't have to go around you. It's your information that you control. Oh yeah, and then the other thing is that when you do genetics, there's often things that you find out about the family that we won't go into also in this discussion. So this is the US specific, right? Obviously this is an international meeting, but the idea is that you cannot be discriminated on based upon your genetic testing. There is a potential of impermanence to this, right? It's just law can change. But generally, this is how we think about it. But other things can be impacted, like life insurance and usually when you have a tumor, they can kind of actuaries figure stuff out. But if there are policies going after kids or things like that, you think prospectively. because once the genetics is known, then you have the obligation to disclose. Okay. Let's see. Okay, so then I think I also want to go through this, but I really like thinking about there being this kind of seismic shift that happened in 2002 when people who were what's called apparently sporadic, so there was no family history. They just had an isolated Pheochromocytoma, it had genetic testing, and then 25% of them had an underlying genetic cause. That number's gone up now, 10, 15%. But what's key here is that when we think about genetics, we're always thinking about like, oh, everybody's affected, everything's really obvious. I would know. I'm 75 years old. There's no way this can be genetic. No one in my family has the tumor. That's not right in this particular condition. Okay, so yeah, and then, okay, so this is like a little bit more data. I'm trying to find a balance between how to display so much information. And what we're looking at here is that these are individuals who are genetically tested, and they were tested across different age groups. So either, before age 10, 20, 30, and what you see here, so let's just look at this one gene, which is VHL, that's in red, that in 10-year-olds, most people have a genetic cause, and VHL is a big part of it. And then up here in the blue, this is the people that you don't find a genetic cause. So we call this sporadic, because we didn't find anything untested. And what you can see is that as you get older and older, it's more likely to be not genetic, it's more likely to be sporadic. That's true. But even in the 70 to 90-year-old folks, you're still finding 15% pickup, right? So everybody should have genetic testing. That's this way of, this is like a graphical way of justifying this. For clinical medicine, the decision in the US that is cut off for doing things is around 5%. If you have a pretest probability around 5%, insurance companies typically will cover that. That's at least historically. Okay, and then this is, again, the composition of the different genes. I think this is too nuanced for our discussion this morning. You see this. Okay, this is, again, one of these big, complicated charts. I just want to emphasize a few things, okay? And so we'll just look at the surface. So genetic testing, why do you do it? Because it can think about not just the Pheochromocytoma Paraganglioma, but other things. I already kind of emphasized that once. It tells you about the inheritance. So who in the family is at risk? And we'll talk about this, what's called maternal imprinting often, but actually a parent of origin. Anyway, that's a small, maybe more my problem than your problem, but we'll talk about what that means. It can tell you what the risks are. Now, if I have a tumor, how is it likely to behave? And it can also tell you about what's the likelihood of my child or someone else in my family getting a tumor. Okay? So it has a lot of predictive and useful information. And I'm sorry, I tried to avoid complicated figures like this. Here, you were actually this was just shown just a moment ago. And I'm not trying to endorse and be data. They're actually, I mean, I won't say anything positive. There's other choices as well. But this is one company that has a panel that's actually quite expansive in its coverage. And what we usually do, and this is relevant for those folks who had they had tested 10 years ago, or they got tested 5 years ago. There are new genes that are now on the panel. So maybe you want to go back to your person and say, are there new genes? Can we look at my old tests? Should I have repeat testing? Sometimes that's appropriate. Also, now we often include what's called RNA testing in a lot of our panels. And what that means is, I told you about the genes, right? Your genes are in your DNA. And when we do genetic sequencing, we read the part of the DNA that tells you about what protein is going to be made, right? That makes a lot of sense. It's part of the instruction manual. It turns out that there's like footnotes everywhere, there's endnotes, there's all these other things that influence how the gene actually works. And in order to find those things, we can't sequence the whole genome necessarily, but you can look at how did the cell actually tell, how is the protein being told to be made? And that message is the RNA. So now we can read that message to say, is the cell putting together the right message? And that's a way of looking at those complicated mutations at the RNA. And then overall, this only adds about 2% genes. But in families where there's some suspicion, and even we typically do it. So I'm not going to go into the molecular biology, but I think the conclusion here is if I've been tested years ago, maybe I go back and talk to an expert and say, should I be rethinking this? Okay. Stephanie, I do have any, I didn't start a clock, I didn't do anything, so just enough. This is a completely, I put slides here, it's untimed talk, so if I'm going too long, just like raise your hand and say, I'm done. Just tell me to stop. Okay, so this is one of those complicated areas. The genetic, we get very clear answers in terms of what the results are when we test your gene, or when we read what your gene looks like. But that doesn't mean we know what it means. We can just read it, right? So interpretation is a whole other thing. And when we interpret variants, so different people have different sequences, in order to say that your variant is associated with or causes your condition, we have to be 90% certain, at least. So likely pathogenic is a 90% certain cutoff, which is pretty high. That means if I'm 89% certain that you have a pathogenic, I tell you you're fine with all. Right? So it's an all or nothing thing kind, right? So, but it's not. The certainty is lower. So your variants of uncertain significance, which sometimes are disclosed, sometimes not, you should try to get your test report so you have that information. Okay, this is a little bit complicated, but it's like all the things that go into figuring out whether or not a variant causes the condition. And there's multiple factors, and they're all summed up in the scoring system. So again, if you have a score of 5, 1 point below likely pathogenic, you're in this VUS category. So you should know that every generation, so every time there's a child, about 100 new changes occur in the DNA. This is a rough estimate. So mutations aren't something that's inherently bad, right? That variation, that's how we change from generation to generation. And so some of those changes can affect the gene in a negative way. But most changes are benign, like English versus English, right? So labor versus labor, that's fine. It's the same. You change the DNA, but it reads the same, has the same meaning. But sometimes you change a letter and it really does change things, right? So that's a change that changes the meaning of the word or the sentence. So I don't want to make you anxious about variants of uncertainty, but understand that there's a range of potential impacts. So you're not necessarily part of this, but in the background, the DNA testing companies like Invitae and some of the other ones that are really well established do a very good job of going back and revisiting variants and figuring out, should this one move category? And they work with us, the researchers, to try to categorize those. But what I would suggest is that if you have a VUS and you have suspicion about it, talk to someone who really knows that gene deeply. The obligation of the testing companies is to run the algorithm, figure out how certain they are, tell you the answer. Our job is to then interpret that and figure out whether or not we should be more suspicious or not. What were the actual points that were given to that particular variant? Okay, this is maybe a little bit nuanced for most people.
Minute.
1 minute time left?
2 minutes.
Okay. Okay, so this is a karyotype, which is showing all your different chromosomes. So you have one that came from mom, one that came from dad, right? These are 2, and you have 46 different chromosomes. And the genes are like located within these little chromosomes, right? It's very, very small. Okay, so what happens when you make an egg or a sperm? You have, let's say you had a mutation that's called SDHB, and you got it from your mom's chromosome. When you make an egg or a sperm, half of those will have that mutation, the other half will not. That's where you get the transmission risk of 50-50. Now, transmitting the gene is very different from transmitting the risk. This is the parent of origin effect. I think with 2 minutes, we don't have time for this. Okay, let me just get these concepts right, because this is something that you should understand well. Penetrance. Penetrance is the risk of developing the condition if you have the gene. 100% penetrance is shown here. Any square has inherited the gene. Green squares actually develop a tumor. So a complete penetrance means 100% of those people develop a tumor. The genes we're talking about are incomplete penetrance. Only a fraction, a subset of people with the gene will develop a tumor. That's called incomplete penetrance. The other thing is that called expressivity. That means, okay, I develop a tumor, but what kind of tumor do I develop? My sister had a neck tumor. Do I get an abdominal tumor? What is it? That's called expressivity. And for this particular condition, what we say right now is just because your brother or your sister had a tumor does not make it more likely that you will. Just because you have one tumor doesn't mean that you're more likely to get another than your brother or sister who didn't have any. So that right now, there aren't like good and bad people, right? That, as far on a statistical basis. Now, this may be revised eventually, but right now, that's what the numbers say. Okay, this is the last thing. And if you inherit an SDHB gene mutation, one of your copies of SDHB has the mutation, the other copy is fine. For the most part, your cells of your body are 100% fine. Every cell in your body is working. I have folks with mutations that run marathons, perform at the highest level of collegiate athletics, do everything. So having a mutation in all your cells does not have real physiologic consequences as far as we can tell. When a tumor happens, there's this event. So for SDHB, you actually lose a whole little chunk of your chromosome. And hundreds of genes get lost. This is the tumor initiating time. So this isn't in all your cells of your body. This is just one cell this happens to happen. And we think when this error happens when you're very, very young. And then it takes decades for it to show up. And that's why I think for the most part, your highest risk is earlier on in life, although it can show up later on in life. And then the issue, people often ask, like, what can I do about this? Well, we think there's not that much you can do about it because it happened, but we don't know, right? So it's not something you did that makes you have the tumor. Okay. I can't go through all of this. I do want to make one final point, which is that we talk about Pheochromocytoma Paraganglioma being rare, and it is relatively rare. But the numbers currently bear out that it's about 1 in 3,500 people, roughly. It's not as rare as people think. We should be thinking about it more often clinically, and we should know a lot more about it given its prevalence. I agree. Yeah. Okay, I think that was enough. So thank you guys very much. Okay.